back, everyone. Next up, we have Bioxytran Inc. It trades on the OTC Pink sheets under the symbol BIXT. It is a clinical stage pharmaceutical company developing platform technologies in the fields of glycovirology, hypoxia, and degenerative diseases to eliminate viruses and prolong lifespan using carbohydrate drug design. Please welcome our speaker today, Executive Vice President, Business Development, Sheikh. All right, Mike, welcome back to the Emerging Growth Conference. It's been a couple of months since you've been on, and so much has happened with Bioxytran. I remember last time you gave a presentation, you indicated Bioxytran was in the caveat emptor status and working on getting traded again. So give us an update, any new developments, and give our viewers a high-level refresher of what your company does.
Anna, I got to tell you, I'm really pleased to be here and report that Bioxytran is trading again under the symbol BIXT. It's on the pinks. And our volume's pretty healthy. We're averaging around 50,000 shares a day, and we're really seeing some good bid support. So I think it's safe to say that since our last conference, our stock is up threefold, and it's very much behaving like an IPO. So it's kind of exciting. Now, on a high level, let's talk about Bioxytran. We're a clinical stage biotech, so we're over that research phase with two exciting platform technologies. Our Glycovirology platform has 2 molecules ready for the clinic and a third under development. We're initially targeting COVID-19 and other viruses. That takes me to our next platform. It's Hypoxia and Degenerative Diseases. Now here we have one literally amazing molecule that's designed to transport oxygen. Now this is still in the preclinical stage, but if we get the proper funding, this could be one of the most impactful molecules developed, as well as the most controversial regarding its use. Now that's a teaser for everyone to stay tuned later on.
We definitely want to stay tuned for that. But one of the things you definitely want to highlight today is your clinical trial results. So please, Mike, go ahead, share your screen, and we're going to follow your lead.
Okay. In our last emerging growth presentation, I presented these slides, so they might look familiar, but I really don't think I gave the perspective that I should have. This first slide represents data from our first clinical trial. Each of those lines, those downward sloping lines and curves, represents a patient's viral load over time. That red line that you see there, the horizontal red line, it represents less than 50 copies per microliter. Now, in the HIV world, 50 means it's undetectable and untransmissible. Now, I want to key in on this idea of transmissibility because right now, the consensus in America is that people think that COVID's over. They can drop their masks, they can do away with social distancing, and COVID is no bigger deal than the flu, okay? That is not true because case fatality rate is 1.1% in COVID versus the historical rate of 0.1% in flu. Now, that means that COVID is still 10 times more deadly and also has the ability to disable people with long COVID. And actually, the updated reports on long COVID tell us that the prevalence is now around 20% of those that contract the virus get long COVID. I want you to think about that. That's a 1 in 5 chance that if you get COVID, you're going to become disabled. And hey, you got a multiplier for the people that are exposed more than once. This isn't a fun disease to deal with. So with these stats, we think it's possible that new endpoints or therapeutics might shift from death and time in hospitalization to time of infectiousness. Now, in healthy people, the virus sheds in around 7 to 10 days. In our clinical trial, the participants were undetectable within 3 days, and that finding was statistically significant with a p-value 0.029. You see, what we're seeing here is that this issue of infectivity rather, is also playing out on the White House setting. Remember, President Biden just got sick, then he got better, the virus rebounded, to the point, and then he got infectious again. All right. So when we look back through that, it took him 15 days to stop his infectiousness. So now what I want people to look at and balance is, do you want 15 days of infectiousness or 3? Which is better? Now, Biden did really well considering the real world data. When we look at that data about Paxlovid, what it comes out to is that 30% of the people, the high risk people that take it, get to undetectable levels in around 20 days. So that's pretty interesting. So let me get to my next chart here. I kind of went off on a tangent there. Now, this graphic here, this shows a viral curve of untreated patients in dark blue. So you see the nice curve that kind of goes up there. You see how it crosses the undetectable red line at 10 days? Now, a therapeutic that reduces viral load needs to go inside of that, and that's kind of where we're at there. So, and I also want to point out that, this clinical trial, it's footnoted, peer-reviewed, the sources in the bottom of the slide. Now, I want to take a big picture. Infectivity, you kind of have to look at infectivity as the holy grail of COVID, because if you can eliminate the infectivity, you're ultimately going to be able to stop the endemic. Now, am I saying we're going to stop the endemic? Heck no, I'm not saying that. And honestly, to prove something like that, you need a ton of funding. And we're not focused on that. We're focused on one thing, and that's regulatory approval. And if we get that regulatory approval, then, that's something for the NIH, Big Pharma, someone else. It's not our problem. All right. Now, the point that I want to drill home to investors is how this one peer-reviewed article that I just went over, okay, has basically de-risked future clinical trials if you look at it in that mindset, because maybe there's some potential regulatory changes coming down the road, and that creates a more challenging regulatory environment, okay? So here we have statistical significance that suggests if you do another study, we're going to see similar results, so that's good. And then we've already jumped over this impossible hurdle, which is infectivity. That's really hard. That's a really high hurdle versus hospitalizations and death. All right. So what I'm trying to say here is no matter how tough the regulatory climate gets, we feel confident we're going to be able to meet the challenge.
So, Mike, let me get this straight. It looks like anyone can enroll in Bioxytran's clinical trials, and you don't necessarily care if they have an underlying disease or what their vaccination status is. So when Paxlovid and Molnupiravir did their trials, they're only enrolling people with at least one underlying medical condition. So it's almost feeling like you're making things harder for yourself than what they need to be. Or is your drug really that good? Because this means you're essentially competing with Big Pharma, Pfizer, and Merck.
Look, we can't answer if our drug is good or not because we haven't done a head-to-head study. So that's but you accurately noted that we were going to recruit almost anyone. There's the parameters listed on our clinical trial. There's a certain CT value. Look, we're definitely not going after the people with underlying medical conditions. We are going after a much larger group, an underserved segment of the population, people like you and me who really aren't in competition with Big Pharma, who don't have these underlying medical conditions. Now, in terms of Big Pharma and the competition, look, we want to work with them, Big Pharma. We want them to license the technology from this. In order to do this, we need to get data that we can present to them, which means completing large clinical trials. Now, when it comes to COVID and getting a deal with COVID, they're not necessarily needed in the picture because what we're seeing is that the governments are actually the buyers, and we don't need them for distribution.
So your drugs are classified as galectin inhibitors. So what are galectins and what do they do? And am I pronouncing that correctly?
Yes, you are. So I got a new slide here. So galectins are proteins that recognize certain types of sugars or beta-galactosides. I'm going to describe them as Pac-Man looking things. If you kind of see, they look like Pac-Man. And they can bind themselves, specific sugars. these galectins serve functions both inside and outside the cell. For drug development purposes, we're going to be concerned with galectins that are excreted and reside in the extracellular domain. Now, think of extracellular domain as outer space, okay? And the planets are cells. That would make galectins like some sort of spaceship that can move around outside the planet. Spaceships can dock with each other. They can make these huge arrays. They can deliver mail from one planet to the next. I mean, I'm taking this to, a kindergarten level here. And then they can help friendly-looking aliens enter through the portal, viruses, right? So that's what galectins are in a nutshell. They're little spacecraft that help make things happen in the cellular universe, okay? So now let's talk about what galectins actually do. So in order to do that, I kind of got to set the stage. There's protein-on-protein interactions in cells where you produce a particular enzyme in the cell. It's responsible for the disease. You block it. Everything's nice, all right? Now, these are easy to measure and easy to develop, or easier to develop drugs. No drug is easy to develop, but now let's juxtapose that against galectins. Galectins are all about the carbohydrate on protein interactions. So galectins have one feature called the carbohydrate binding domain, CBD, but not the CBD you're thinking of, right? And this Pac-Man looking spaceship wants to only bind with sugar. So when galectins bind in different ways, they bind to themselves. You kind of see on the bottom there of the slide the different ways that they can do that. Galectins act as signaling molecules. They perform a wider range of functions outside the cells by binding to multiple targets and multiple proteins. Now, in the hierarchy of the cellular universe, galectins have diverse functionality. Certain subtypes of galectins are overexpressed in many diseases, like galectin-3, and have become important druggable targets. Here we can see an example Here we can see an example of galectins, okay? They can modulate signaling. They can go through the receptors. They can bind receptors, different kinds of cells. They do a whole bunch of things there down at the bottom. And hopefully my galectin spaceship analogy kind of makes sense now.
It does. I need it on that kindergarten level for this type of technology. So now, Mike, there are a few medical journal articles. They've linked high galectin serum levels to COVID deaths. So how good of a predictor of mortality are these biomarkers? And do you think your galectin inhibitors could stop COVID deaths?
I put up a slide there so you can kind of see what we're doing. We found a couple of journal articles that measured the correlation between the high levels of galectin-3 and galectin-9 mortality. There's something called this Roc curve, and that's depicted there. And it plots a trade-off between a biomarker that can predict death versus incorrectly predicting life, okay? The area under the curve is measured on a percentage basis. So the ideal is 100%. It's very predictive versus 50%, which is that red line. That would be 50%. Galectin-9 is 98%. Galectin-3 is 91% predictive of mortality. I want you to let that sink in. Now, these biomarkers are highly predictive, but what I want everyone to realize is that galectin-3 serums almost perfectly describe people with underlying medical conditions. When I first heard of COVID, my mind immediately went to these people with underlying medical conditions who weren't faring well. I was like, I was wondering what are these galectin-3 serums like? I was dying for them to do a study on it. Finally, we got one, okay? But galectin-3 is elevated in people in diabetes, hypertension, heart disease, dementia, asthma, and that fits the classic definition, CDC definition of underlying medical conditions. Now, in early stage disease, there's clinical evidence that galectin inhibitors can stop the viral load. Now, while we have little evidence of galectin inhibitors being used in late COVID, it's completely reasonable to think that they could prevent death. Out there right now is an inhaled version of a galectin-3 inhibitor called GB0139. It was tested in combination with another drug and demonstrated changes in efficacy, markers of hospitalized COVID-19 patients. So there's a little bit of evidence there in this late stage disease. And based on this data and understanding of our molecule and how it's more likely to stay in the blood more efficiently than an inhaled drug, I think there's a good chance that galectin inhibitors could really actually help people cheat death.
You're blowing my mind here. Let's lighten it up for a second. I hear you sometimes play games with investors and ask them to take the galectin challenge. Sounds like a good game show. So what's the galectin challenge? How do you win? And do you need a host?
You heard about my galectin challenge. Well, whenever an investor asks, so just how big is this galectin technology? The odds are this is the first time they ever heard of the word galectin. Mm-hmm. So I just can't say, oh, it's involved in hundreds of diseases, because then people can't really relate to that. And honestly, most people couldn't name 100 diseases if they even tried. I kind of have to take them through a process of searching in Google for disease indication plus galectin-3. So the galectin challenge is to basically do a search and not find not to find journal articles on it. So you would type in galectin-3 and any chronic disease, and you're going to come up with journal articles. The idea is stumping me and not finding it. So that's how you win. Stump me. So stump me with the most outlandish chronic disease indication that you can conjure in your mind. And if you investors want to play this at home, do it right now. Open up your browser, look at the most craziest chronic disease out there, type in galectin-3 and see what happens. And do more than one. It'll blow your mind. So you want to play the galectin challenge? You want to challenge me, Anna?
Mike, I'm going to take your word for it. You seem to know what you're talking about. So no, I'm good. Thanks.
All right. So here, let me, I want to get a new slide up there. If you look at this slide, galectins show up as biomarkers of chronic disease across key organs. Look at the research, and it suggests galectins are ideal markers of disease. Over the years, these journal articles have built up like this huge mountain of evidence that galectins are responsible for these chronic diseases. Now, what's missing in these journal articles are drugs that target galectins. So we think this is a great idea, but someone needs to actually make a drug. And that's what we're doing at Bioxytran. We're taking this massive amount of evidence and translating it into action and targeting these galectin biomarkers, which are thought to be drivers of disease. And the common thread across all these studies is chronic disease.
Well, Mike, you sound like you've been studying galectin science for some time now. I'm personally curious what your background is in. And we did a little digging, found out you wrote a short paper about the galectin effect a few years ago. So is this galectin effect good or bad for people?
Well, I've been self-taught, Anna, all this time. And when I talk about the galectin effect, a picture's 1,000 words here. So I put up this graphic because it'll really help. The galectin effect relates to the functionality of your T cells. T cells are your white blood cells, and they're the fighters of infection and viral diseases. And T cells are used in cancer and virology. And when they misfire, like in this graphic, it's called T cell energy. Okay, so when these white blood cells or T cells operate normally, what they do is they latch onto the target cell without, with their little grappling hook receptors. So they got these receptors and they kind of bring it close to them. That's the picture on the right side. That's how it's supposed to be done, right? So, and then what happens is you see that little green stuff that punches holes there, and the green stuff is, the cytotoxins, and it flows through and kills the other cell, and keeps it within the cell membrane. This is how T cells normally kill. They're highly selective, but susceptible to this galectin plaque. And you can kind of see that galectin plaque, it's kind of little red stuff, those are that represents the galectins. So once you have the plaque, it forms around the T cells. The cell becomes energetic. It doesn't mean it's dead cell. It just means that it can't kill as effectively. And you see this T cell energy in this, it's called the CD4 to CD8 ratio that the doctors look at. And it's a proxy of your overall immune system health. So the galectin effect disrupts the immune system in pretty much the same way it does for both viruses and cancer. The bottom line, the galectin effect, it's not good.
So you went over one journal article, but it looks like you had a couple other recent releases. Can you summarize the significance of the other ones?
Sure. First let me talk about the reaction to our journal articles in the scientific community. It's been great. Okay, they seem to understand. Bioxytran has come up with this methodology to create carbohydrate drugs that treat the entire disease instead of just chasing after these little singular targets. Okay, you hear about these targets like IL-4, IL-6, TNF alpha, CCR5, VEGF, CD14, PD-L1. The list goes on and on. With this discovery, we've taken drug design, at least in carbohydrates, to the next level. We're talking about next generation galectin inhibitors. Instead of creating a molecule to just target one receptor, why not target multiple receptors? That kind of makes sense. You have all these scientists that you see on a whiteboard, they start scribbling, this goes up, this goes down, this goes over here, that goes over there, and they're talking about pathways, right? Well, all those up and down, left and right, that's a disease signature, okay? So now let me pick on galectins and kind of put this in a ball for you so you can understand. What if we had a disease where galectin-1 was upregulated and galectin-9 was upregulated? We would use nuclear magnetic resonance, NMR, to screen these molecules. I'm not going to get into what it is because it's nuclear medicine, right? But the molecule, but then what we do is we look and try to design a molecule that would inhibit both in one molecule instead of doing one molecule to do galectin-1, another molecule to do galectin-9, right? And we don't have to do animal studies. That is the power of this discovery. And I don't think people are getting it. So there's a multiplier effect in creating new molecules. Now, let me give you a little bit of a history lesson so you get it. The first galectin inhibitor was invented in 1994. The next one was in 2001, and another one in 2008. And by the way, all three of these were invented by our CEO, Dr. David Platt. And then he took a little bit of break, and then one of our competitors, Galecto, invented TD139 in 2011. Now, from 2011 to 2020, there were no new galectin inventions. Now, BIXT, Bioxytran, has 3 more galectins now, targets, and galectin has 1 more. So in the past 2 years, I want you to think about it. We've created more galectin inhibitors in the past 2 years than in the past 18 years. Now, knowing how to apply this technology is a major competitive advantage, and investors haven't seemed to grasp the significance of this yet, which is why I'm coming on here to present. Now, I've also interacted with a couple of investors, and they told me the journal articles, they're technical, it's hard to understand. Yes, that's why you have me, a galectin evangelist, okay? And I'm going to translate this for you so you can understand the science in little bite-sized chunks so you can understand it. The bottom line is that we're leaders in glycovirology, and we have a major competitive advantage in technology. And hopefully that's what investors get out of this. Now, the other journal article, I'll just kind of go over this briefly. It blocked the galectin. It showed that we blocked the galectin-3 and the activity of the virus to infect cells. All right. And I want to throw that word infectivity out there, like I did with President Biden, and how we believe we can stop it quickly. So that means the journal article basically gave us bragging rights that we have a galectin antagonist because, there's more than one out there. Okay. But now I want to compare these galectin molecules that were the new generation of lectin molecules that we're developing. I'm an Air Force guy, so I like analogies, okay? So if molecules were airplanes, okay, we got a fleet, 5th generation fighters on our hands. And I cannot wait to see how they perform in combat, okay?
This just gets more and more fascinating. So after hearing your story, I'd like to actually hear more of your story. If you've been in the Air Force, my goodness. So it's really hard for me to fathom that big pharma isn't just knocking at your door, trying to throw money at you. So I don't understand why stories about your company aren't plastered all over the news and internet. There's such a promising story no one's talking about. So what's the disconnect?
Yeah, thank you, for pointing that out. It's actually amazing. There's over 4,000 journal articles written about galectins and all the things that they do. So yeah, scientists know about galectins, but there's a scarcity of companies doing galectin research. If you think about it, there's only 3 public companies out there that have developed galectin inhibitors. And 7 of them, it's approximately 7 of them have made it to the clinic. And then you have Dr. Platt sitting over here, and he invented 5 of them. So the reality is you only have a handful of scientists, and I'm being generous at a handful, who have the actual experience to formulate a galectin inhibitor and manufacture it. This scarcity of talent means that there's no one out there in big pharma for us to talk to. I'll give you an example of what it's like. There was a revolution of monoclonal antibodies to target disease, and it happened in very much the same fashion. There's a few small companies developing the technology for a decade, and eventually big pharma recognized them all at once. And then they went on the acquisition binge, and we have a whole bunch of monoclonal antibodies all over the place, right? Okay, so this is why we need to get more data and more data quickly. And peer-reviewed data, because otherwise no one's going to believe us. I mean, look at our first clinical trial, complete viral elimination. I wouldn't believe me either, okay? So now look at Pax. That's why we got peer-reviewed. So I believe me now because it's peer-reviewed. Now look at Paxlovid. That's the biggest drug on the planet. It needed 15 days to clear the virus from President Biden. So people are going to look at you and say, wait a second, how does a small little company have a solution that could possibly change the complexion of the pandemic when tens of billions of dollars were spent on these vaccine solutions and these therapeutics? There's a disconnect there. Mm-hmm. And the only way to bridge that is with more data. We have to get the peer review done. We got to get the scientists buy-in, talking about it. And then eventually it's going to make it in the news. This is the normal order of things. And we got to stick to that thing. We can't get ahead of ourselves. We can't get over our skis. Look, we live in a show me world, and data is all that matters. When we finally do release the data, we won't need any fanfare because the data is going to do the talking for us. I encourage investors to take the time to understand the concept of statistical significance, because then you're going to be properly risk rated in this position.
Well, someone might lose a lot of money once this data's out there. Anyway, so you mentioned 2 platform technologies, and most of our conversation, it's been about the Glycovirology platform. So we are running short on time a little bit. I want to make sure we get some more questions in. Tell us a little bit more about other platforms and how you plan to develop it. And don't forget to tell us why it's controversial. You teased that in the beginning.
You remembered. All right. BIXT25 is our oxygen transport compound, and it has broad applications, but we're primarily developing BIXT25 for the treatment of ischemic stroke. Before we get into how we make it and what it does, let's do a review of how blood works, because otherwise you're not going to get the connection here. When blood circulates throughout your body, it eventually releases oxygen trapped in the red blood cells. I mean, we know this happens, but let's get deeper. So what happens is the oxygen concentration in the surrounding tissues has to be lower for that molecule to diffuse there. All right. So, and our blood runs every 45 seconds. So think about how many trips it makes and how much it does. Many people don't realize that the release of oxygen is based off of physics and not a biological or chemical reaction. So I want to make sure that we got that there. So Now the magnet, and again, I'm dumbing this down, the magnet that keeps the oxygen in tow is the heme molecule, the heme in hemoglobin, hemoglobin, right? It's the heme. So the way we make BXT25 is we stabilize heme. The red blood cell molecule that holds oxygen in a carbohydrate scaffold, right? Now, this protects heme from degradation and allows it to circulate and carry oxygen longer, slowing its breakdown. The drug is an IV formulation and needs to be circulating in the vasculature to work. So you can't just like take BXT25 as a pill. So our first indication that we're targeting is ischemic stroke. And by supplying oxygen transport to these patients, we're going to keep their brain alive while the blood flow is restricted. Our molecule is 5,000 times smaller than red blood cells. So BXT25 passes through the clot like water goes through a fishnet, right? The idea behind that is that within 15 minutes of the time that it takes for a paramedic to arrive, he's going to be on site. He's going to say, "Ooh, he's going to look at you and, then realize you have a stroke." And then during the stroke, the brain is pretty much holding its breath. So as soon as the paramedic confirms that it's a stroke, it puts, and this is all hypothetical, it puts the IV in, he'd administer the BXT25, and then what we're hoping to see is a reversal of the obvious stroke symptoms shortly after administration. So then the patient gets a transport to the hospital, gets their imaging, and figures out if it's an ischemic or a hemorrhagic stroke. Now, oxygen is a basic part of all cellular processes. If you hold your breath for a minute, people are going to figure out how important this oxygen transport molecule is. And now I'm kind of getting to the teaser, okay? Many people out there are looking for hyperbaric oxygen treatment to clear their maladies. And whether it's stroke, dementia, Parkinson's, they're using these things for wound healing. They're using these things. Now, the challenge with hyperbaric oxygen is that people can only get 1 hour. Why? Because extended use means that they could have a breakdown of their organ lines. That's like one of the side effects. And it's not like it's going to break down right away. It's extended usage. Okay, so I don't want people to think that hyperbarics are bad. Now, the truth is, let me go back a second here. One little dosage of the BXT25, or maybe a double dose of BXT25 could be the equivalent, we calculated theoretically, of around 18 hours of HBOT treatment. So I want you to think about that. If you had 18 hours of HBOT treatment, I don't know what we could do with the human body right now. You're getting 100% oxygen 100% of the time. I have no idea what can happen. So, and this is the teaser. So approval of this drug is going to create controversy. You got athletes that train at altitude and try to get an advantage by training their blood to deliver more oxygen. They just need this in their system. So because at altitude, their body genetically reprograms its blood cells to carry more oxygen. And here you just have it at will, okay? So it's the same concept of how Sherpas can live at 20,000 feet. So there's ethical considerations for sporting events and all these things that could come into play. I mean, think about it. Imagine sprinters that never get tired. I mean, there's going to be world records set all over the place if this is allowed in competition. And I don't think it should be, but anyhow, that's the teaser.
This is some Superman technology you're talking about now. Okay, we've got about 10 minutes or less. So I want to get right into investors. Why should investors buy your stock?
Well, there's the fundamental reasons, and then there's the technical reasons. The fundamental reasons I talked about. So there's no sense in rehashing all the fundamental reasons. So let's talk about the technical reasons. Look, we have a thin float out there. We got a great share structure, 101 million shares out there. But I think it's 145 million if it's fully diluted. There's no like large warrant overhang. OTC investors, I know that's what they're looking for. you have huge insider ownership, So, there's something to say. Management has skin in the game. And, by the way, we have forfeited millions of dollars over $1 million in payroll debt because we believe in the company. And I think we got a winning management team. Because we didn't talk about that, but I think we're a winning management team. We got the Dr. Platt with over 30 years of experience. We have a potentially quick approval pathway by going after COVID. COVID isn't cancer. COVID, you're over it in weeks, not years like cancer. I don't know why I use that example, but I'm not saying we're going after cancer. You also have licensing and label expansion strategy. That I didn't really talk about that, but once we get it approved for example, for COVID-19, we could expand the label very quickly. And this point I want to beat home is best of breed technology. We have best of breed technology.
So let's talk a little bit about your recent quarterly filings. You just released that. There's some new developments with respect to the pipeline. Can you take a moment to update our viewers on that?
Yeah, there's some updates in the pipeline. I encourage you to just go to the filing. I don't remember what page it was on, but yeah, we got some updates to the filing. I'd have to have it in front of me to go over it, okay? But
Sure.
definitely take a look at that. And I think there was also an 8K today. We did an basically an at the market financing, and I think that that goes to show the power. It goes to show how investors really believe in us to do that. Really wasn't at a discount. It was at the market. So that speaks volumes that we can get a big player like that.
And Larry Bonafide wants to know, what's the name of your COVID-19 fighting drug?
Oh, I can't believe we didn't talk about it. It's called Prolectin M. Prolectin M. Thanks, Larry.
All right. Well, Mike, would you like to say any closing remarks? I know we have some stories, Beth's story, if you want to end with something like that.
No, I hope what this presentation does is I hope investors walk away with a better understanding what exactly the Bioxytran opportunity is. We're myopically focused on drug approval. The other thing that I want to impart, we're a fiduciary of your money. We're doing things, we're operating very efficiently. We have a very efficient design process. And, just keep that in mind. We're going to be good stewards of your money. And then I got to plug social media. Don't forget to follow us on social media. Go to our website, subscribe. We need that. We're going to be communicating on social media. You'll find out my Twitter handle is @MikeSheikh2. So please follow me. I want to see lots of follows. I will respond to your questions on Twitter. Yeah, and I look forward to the next conference.
This was a great presentation. I think we all got an advanced biology and virology lesson today. And I thank you for your thorough presentation and this important work you're doing out there. So yes, join us again in the future. We want to hear your updates for sure. Thank you, Mike, for joining us again.
Thank you. I'll come back.
All right, everyone. What a great day it's been so far. We are not done, so stay with us. We're going to come right back with our next presenter in just a few minutes.